All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria.
We learn the different parts of a cell and their corresponding functions. And sizes are related to the functions of the cell. The basic differences between most plant and animals are their shape, size, and the absence and presence of an organelle. But before we discuss their differences, let us first talk about their similarities. Both plant cells and animal cells are eukaryotic. This means that plants and animals are multicellular, and they possess a true nucleus, a membrane-bound nucleus. The basic structures of eukaryotic cells are cell membrane, cytoplasm, and nucleus. Both plant cell and animal cell contain the following organelles: mitochondrion, ribosome, Golgi bodies, nucleolus, vacuole, lysosome, and endoplasmic reticulum. Let us now talk about their differences, starting with their shapes. Observe the pictures of animal and plant cell. What do you think is the difference between them? Animal cells come in various shapes and tend to have round or irregular shapes. On the other hand, plant cells are more similar in size and are typically rectangular or cube shaped. When it comes to size, animal cells are generally smaller than plant cells. Animal cells range from 10 to 30 micrometers in length, while plant cells range from 10 to 100 micrometers in length.
Cells. Beginning with cell walls. In plant cell wall is present and is composed of cellulose, whereas in animal cells, cell wall is absent. The shape. Plant cell is rectangular and has fixed shape, whereas animal cell is round and irregular in shape. Vacuole. It has one large central vacuole taking up 90% of cell volume and is filled with watery fluid, whereas animals have small vacuoles, much smaller than plant cells. Chloroplast. Plant cells have chloroplast because they make their own food, whereas animal cells don't have chloroplast. Plastids. Plastids are present in plant cells, whereas plastids are absent in animal cells. Lysosomes. Lysosomes usually not evident in plant cells, whereas lysosomes occurs in cytoplasm in animal cells. Centrosome. Centrosome is absent in plant cells, whereas centrosome is present that helps in cell division in animal cells.
Two-kingdom system of classification. Carl Linnaeus proposed two-kingdom system of classification. He classified all organisms into two kingdoms, that is, kingdom Plantae and kingdom Animalia. Carl Linnaeus is also known as the father of taxonomy. Not only this, but he also made great contributions in the fields like binomial nomenclature for naming plants and animals, taxonomy, taxonomic categories, and much more. To solve this problem, Ernest Haeckel, a German biologist and philosopher, created a third
Except for
kingdom
sex cells,
called
such as
Protista,
sperm and egg cells.
where he located all unicellular organisms. This classification is also known as Three Kingdom System of Classification. Herbert Copeland proposed a new kingdom Monera, in which he placed bacteria, since they are all prokaryotes. This is also known as Four Kingdom System of Classification. Before we take our system of classification, bacteria and fungi were placed in kingdom Plantae. As more and more characteristics of fungi and other microorganisms became known, more advanced classification became the need of time. Kingdom Fungi was given by R. H. Whittaker in 1969 on the basis of mode of nutrition. He proposed five-kingdom system of classification by considering the criteria like cell type, body organization, mode of nutrition, type of reproduction, and phylogenetic relationships. Carl Lewis proposes the modification of the Monera kingdom by finding that bacteria have different types of metabolism. He proposes dividing them into two large groups, which he named bacteria and archaea. He transformed the Monera kingdom into two domains, that is, bacteria and archaea. Since archaea and bacteria have not been subdivided, both domains and kingdoms can be considered. This was proposed by Woese in 1977 when he was noting differences between archaea and bacteria present at the level of ribosomal genetics. He kept rest of the kingdoms together in one domain, that is domain Euchera. This system of classification is also known as three-domain system of classification or six-kingdom classification.
The cell cycle describes the sequence of cell growth and division. A cell spends most of its life in a state called interphase. Interphase has three phases: the G1, S, and G2 phases. Interphase is followed by cell division, which has one phase, the M phase. Together, these four phases make up the entire cell cycle. G1 of interphase is sometimes called growth 1 or gap phase 1. In G1, a cell is busy growing and carrying out whatever function it's supposed to do. Note that some cells, such as muscle and nerve cells, exit the cell cycle after G1 because they do not divide again. A cell enters the S phase after it grows to the point where it's no longer able to function well and needs to divide. The S stands for synthesis, which means to make, because a copy of DNA is being made during this phase. Once DNA replication is complete, the cell enters the shortest and last part of interphase called G2, also known as growth 2 or gap phase 2. Right now, it's enough to know that further preparations for cell division take place in the G2 phase. Now that interphase is over, the cell is ready for cell division, which happens in the M phase. The M phase has two events. The main one is mitosis, which is division of the cell's nucleus, followed by cytokinesis, a division of the cytoplasm. So at the end of M phase, you have two daughter cells, identical to each other and identical to the original cell. Let's review. The cell cycle describes the life cycle of an individual cell. It has four phases, 3 in interphase and 1 for cell division. Most cell growth and function happen during G1. The cell enters the S phase when it needs to divide. In this phase, the cell replicates its DNA. Replication just means the cell makes a copy of its DNA. In G2, the cell undergoes further preparations for cell division. Finally, we have cell division in the M phase. The M phase consists of mitosis, which is nuclear division, and cytokinesis, or division of the cytoplasm. We'll explore the details of mitosis and cytokinesis separately.
Development is the process of progressive and continuous change that generates a complex multicellular organism. It starts off with the gametes, where the sperm fertilizes the egg through a species-specific process. This creates the zygote, a fertilized egg with a diploid chromosomal complement generated by the fusion of the haploid male and female pronuclei. After fertilization, the zygote goes through several developmental phases to create an embryo. The embryo consists of a developing organism prior to birth. In mammals, the embryo refers to the early stages of development until the end of the organ-building phase, known as organogenesis. After this, the developing mammal is a fetus until birth. At birth, it becomes a neonate. And it doesn't stop there. Development progresses to the maturation of the adult form and continues into senescence.